Hello students, welcome to EPG Pathshala. I am Dr. Deepak Kumar. I am Assistant Professor in Department of Biotechnology and Molecular Medicine in Pandit B.D. Sharma Postgraduate Institute of Medical Sciences, Rohtak. Today we are going to discuss about the module Production of Vaccines and Pharmaceutical Proteins. It is under the paper Animal Cell Biotechnology. The objective of this module that what are the vaccines and what are the types of vaccines, how they are produced, and we will also discuss the pharmaceuticals, and we also discuss the role of biotechnology in the production of these vaccines and the pharmaceuticals. So first is, what is vaccine? Vaccine is a biological preparation that improves the immunity against a particular disease. Sometimes what happens, you get exposed to a infectious agent, and you it may cause some serious side effects, and it's also create disease. And so the thing is that if one can get a vaccine that improves the immunity, if your immune system is already activated, then it's kind of preventive measure. So that is a vaccine. So it's a biological preparation that improves your immunity. The vaccines typically contain an agent that resembles the disease-causing microorganism. So the vaccine is something that if a particular disease is caused by a microorganism, like a polio vaccine, the poliomyelitis, which can cause polio, if you can have, it should be resemble with that one, so that it can produce against that microorganism. The thing is, it should be resemble the disease causing microorganism, and it made from a weakened, that microorganism should be weak or in a killed form of a bacteria or any toxins, because if it is a killed form, then it lose its virulence. It will not cause the disease, but it will activate your body's immune system. What happens if the white blood cells in your body, so the antigen presenting cells, B cells, T cells, recognize and destroy the antigen, and they remember that version of pathogen for future. So later, whenever you get infection from that particular antigen, your immune system quickly responds to it and destroy the version, and you are safe from the infection. So the term vaccine and vaccination are derived from the this variole vaccine, a smallpox of a cow, and it is devised by Edward Jenner. So how do vaccine work? The human immune system is a system of biological structures and the processes, and due to these processes, it protects us from the disease. Because it can recognize the germs that enter in our body as a foreign invaders, and these invaders are referred as antigen, and these antigen are termed as antibody generator, because by the reaction of this antigen, the immune system generates the antibody against that antigen. So what happens when antigen invade the human body? The immune system respond to that antigen and produce the protein substances. That protein substance is called the antibodies, and these antibodies are highly specific cells, and these specific cells fight with the invading germs. So when a sufficient number of antibodies have been produced by the body to fight against disease by the immune system, so that provides the protection against that particular germs. It can be for a month, even for a year, even for the lifetime. So even by one dose, you may not be get protected, but by number of doses, 2 or 3 doses, or by the booster doses, one can be protected for the lifetime. So once you are immunized, then a person, when later come in contact with the same pathogen again, the immune system will able to quickly reproduce the same type of antibodies. That's why it is preventable for the disease from developing or decreasing the severity, and is also eliminate the pathogen from the body. So once the pathogen is eliminated, then you are safe from the infectious agent. So though the immunological memory, we can expect that the immune system can remember or can counter about 1,000 or millions of different microorganisms or different organisms. So that's why vaccine is very effective. So vaccination involves the introduction of limited quantity of specific disease antigen into the human body, which stimulate the immune system just enough to produce the amount of antibody needed. There are types of vaccine. There are different types of vaccine, and these types of vaccine, they are classified on the basis of their preparations, that how these vaccines are prepared. To understand the production of vaccine, one should understand the type of vaccine, because different types of vaccine are produced in different way, like live attenuated vaccine, second is killed inactivated vaccine, third one is a subunit vaccine, fourth is a toxoid vaccine, conjugate vaccine, DNA vaccine, recombinant vector DNA vaccine, recombinant vaccines. These are the different kind of vaccine, and they're produced in a different by using a different procedures, and that we will discuss now. So first type of antibody is live attenuated weakened vaccine. This vaccine contains a modified strain of pathogens or bacteria, viruses which has been weakened, and but it able to multiply within our body and remain antigenic enough to induce a strong immune response. The thing is, we have to weaken the strain by passage or by heating. By number of passages, the bacteria or pathogen lose its infectivity, but it's able to produce the antigens. So the examples are the varicella zoster vaccine, oral polio vaccine, or yellow fever virus vaccine. Sometimes the first time the vaccine was produced, it is a attenuated vaccine produced from a strain that are pathogenic in animals but not in human, like cowpox vaccine. Cowpox vaccine is the first vaccine that cause infection in the cow but not in the human being. So the cowpox vaccine that protect against the smallpox in human. Second type is the killed inactivated vaccine. To produce this kind of vaccine, the bacteria or virus are killed or inactivated by chemical treatment or heat. The most commonly used chemicals are formaldehyde and the chelatic agents such as ethylene oxide and ethylamine, etc. So what the reagents do? These reagents cause cross-linking in the nucleic acid chain, and that's why it inactivates the microorganisms, but it does not alter the protein. So that's why the antigen is still present and it can produce the antibody. So the examples are inactivated polio vaccine, poliovirus vaccine, tetanus, pertussis, and rabies vaccine. Subunit vaccine. This type of vaccine contains a very small part of microorganism that is selected for ability to initiate the specific immune response. It means we have not to infect with the or inject the complete virus like we are using in the live vaccine or in this killed vaccine, but in this vaccine we are using a very small part of the microorganism, but that part should be should have ability to induce the specific immune response. So that is called a subunit vaccine. The examples are Haemophilus influenza type B vaccine and pertussis vaccine, the cellular pertussis vaccine. Toxoids. Toxoids are the important subgroup or subunit vaccine that are produced from the microorganism inactivated toxic compounds. The immune system on infection by the pathogen from antibodies that utilize the corresponding bacterial toxin is stimulated. So once the toxin is injected, that acts as an antigen, and that toxoid against that toxoid the immune system develops the antibodies. The examples are diphtheria toxoid vaccine, which contain a chemically modified bacterial toxins that are retained and that toxin retain its immunogenic property. Conjugate vaccine. A conjugate vaccine is created by covalently attached to a poor antigen to a strong antigen, that is thereby eliciting a strong immunological response to a poor antigen. Because sometimes the antigen is poor and it's not able to produce, induce the immune system. So that's why we need a conjugate, so that these conjugate make a stronger the vaccine to make a stronger immunological response. Most commonly the poor antigen is a polysaccharide that is attached to a strong protein antigen. The example is Hpv vaccine. DNA vaccine. What is DNA vaccine? It involves the direct introduction into the appropriate tissue or a plasmid containing the DNA sequence, encoding an antigen against which an immune response is sought. So it relies on the in situ production of the antigen target. Means that a plasmid which contains the encoding gene is injected, and the plasmid produces the antigen, encode the protein, and then act as an antigen. So it's all in the body. The example is the influenza vaccine, but as of now, I think there is not a vaccine which is particularly is in the market of this type, which is approved. The recombinant vector vaccine. In recombinant vector vaccine, the inserted genetic material causes the bacteria to display the antigen of another microbe. So we have there is a bacteria which is not harmful, the bacteria which is harmless, and we make that bacteria with the harmful microbes to provoke an immune response, and the carrier virus that carry the microbial DNA to the cell. The recombinant vaccine closely related to the natural infection, and therefore it is good. It's good to stimulate the. It can do a good job to stimulate the immune response. Examples are recombinant vaccines. Vector vaccines for HIV, rabies, measles are under development. Recombinant vaccine. Recombinant vaccine is little different than the recombinant vector. In that way, it involves that the inserting of DNA that encode a antigen, and that stimulate the immune response into the bacterial or mammalian cell, or expressing the antigen in this cell. Then so we have to first in the cell culture system we have to do this process, like in a vector we have to we have to clone a DNA coding sequence which encode for the antigen, and then we have to purify it. I will discuss later about this thing. So what we have to do, we have to first express it in the cell culture system, and then we have to purify it. So after purification, this antigen can be used as a vaccine. So most of the vaccine these days under investigations are based on such purified recombinant proteins or subunit of antigens. Examples are hepatitis B and human papilloma virus. Saponin as vaccine adjuvant. As I talk just now that there are some vaccines which show poor response, so we need to add something. So that is the adjuvant. The saponin is a substance which is a natural glycosides of steroid which exhibit many different biological and pharmacological activities. So saponin can activate the mammalian immune system, which leads to the significant interest and their potential as vaccine adjuvant. So what is the principle of vaccine production? Standard manufacturers use a bacterial or a viral antigen. Like we need the bacteria and viral antigen it should be very specific, and the strain should be very clean. It should not have any kind of contamination. It's a very clean and a very specific strain one has to identify. So what the manufacturer do? The standard manufacturer use the bacterial viral antigen, and which may be killed or may be living but attenuated. And to make a attenuated vaccine, the disease causing organism is grown under special laboratory conditions so that it can lose its virulence or disease causing properties, so that the vaccine does not cause any disease. And the attenuation can be obtained by heating or passage of the virus in foreign host such as embryonated eggs or in tissue culture system. So the cell culture are required for viral vaccine, since the viruses can replicate only inside the living cells. For example, to produce a polio vaccine, attenuated was only achieved with high inocula and rapid passage in the primary monkey kidney cells. And the inactivated vaccines are produced by killing the disease microorganism by chemical or heat. The question comes, what is the complete process? Okay, one thing like inactivation. After inactivation, we can create a vaccine, and second thing, after inactivation, we have to purify, and then we have to we can create the vaccine. Thing is that first thing we have to start with the culture. Whatever we want to, we have to start the culture of that particular microorganism or a germ. Either we can after culture we can either do inactivation and purification to do the vaccine, and we can also in the culture, after culturing we can also do the antigen purification, and then we can inactivate that, and then we can also prepare the vaccine. Another thing, attenuation, and then culture, and then we prepare the vaccine. And another one is the cloning. We have to do the cloning of particular antigen, and then we can culture it, and then we can prepare the vaccine. So vaccine, as I told, that vaccines are currently these days are mostly produced by the gene techniques. Instead of using a virus or bacteria, a single cell, usually this is surface glycoprotein of the virus, that is exposed to the this is a part of the virus that is exposed to the immune system. So we can express it in the foreign host by cloning. So expression vector, we can make large amount of antigen as used in the vaccine. So these are the current day vaccine. This is the most important to understand. So most used bacteria vector for expression these days are like bacteria, E. coli, yeast, baculovirus, and this process induces the vector to produce antigen, and which can be then later on purified. Now these purified antigen, which can be combined with the adjuvant and is safe and a very effective vaccine, like the human papillomavirus vaccine, which prevents the cancer, and hepatitis B vaccine. First thing is we have to identify the microorganism, that which microorganism we need, and as I told, that the strain should be highly purified and very specific. It is not like, it should not be contaminated, it should be very specific. So first of all, we have to identify the strain for the vaccine production. Second thing, we have to grow that strain into the culture. So we have to grow that microorganism in the culture. Once we have the strain in the culture, then we can isolate and purify the microorganism. So third step, first step we have to select the microorganism. Second, we have to grow in the culture. Third, we have to isolate and purify the microorganism. Once we purify the microorganism, then the process which I discussed, like inactivation or the killing of or like the other thing, like isolation of is antigens after that is come. So fourth part is inactivation of organism. We have to inactivate the organism so that the organism loses its virulence, and after that we have to formulate the vaccine as discussed, as we will discuss. And once we formulate the vaccine, then we have to check the quality control, and then it can be packaged in the lot and it can be released worldwide. What are the other pharmaceuticals that can be produced? So there are certain other pharmaceuticals which can be produced by recombinant DNA technology, like recombinant hormones like insulin, growth hormones, follicle stimulating hormones, blood products like albumin, thrombolytics, fibrolytics, and clotting factors like factor 8, 9, tissue plasminogen activators, and recombinant hydrogen. We can also produce the cytokines and growth factors as well, like interferons, interleukins, like colony stimulating factor like interferon alpha, beta, gamma, erythropoietin, interleukins, and the other stimulating factors. The monoclonal antibodies we can also produce, like using the mouse dynamic or homogenized whole molecular fragment, single chain, or bispecific, like, and the conjugates also. We can also prepare the recombinant vaccine as I discussed. Recombinant enzyme, dynase, also listed some more here. Other products like BMP, protein antibody, regulated recombinant protease antagonist as well. So you see there is a complete list that the human protein which can synthesize by gene cloning in the bacteria or in the eukaryotic cell, which can be used as a pharmaceutical product, like antitrypsin and factor 9 and interferons, TNF, serum albumin. There's so many products which can be used, using a recombinant DNA technology. Okay, so I would like to just give you the little brief overview of the recombinant DNA technology. What is that? First, we need a vector, and we also need a gene of interest. We should know what we want to clone. Right? like we want to clone a encoding sequence for the vaccines antigen, or we want to incorporate the encoding sequence for any kind of hormones or any kind of other biological product. So one should be have a little bit a good hand on these biological molecular technologies. Like for the PCR, by PCR you can pick up your encoding sequence. You have to also make the overhang with the restriction enzyme, and so that you can use those overhang restriction enzyme to ligate your gene of interest into the vector. So in that way, when you pick up the gene by PCR, and you also make a cut with the restriction enzyme, and then you can do the cloning in the vector, and make sure that your gene of interest should be in the correct frame, so that the protein which is produced, it should not be a truncated protein. It should be a functional protein. For functional protein, it should be in correct frame. Your coding sequence into the correct frame. So that just this is a point to be noted while whenever you do this work. So after you cut down the vector and your insert, you can do the ligation, and then you can transform into the bacteria. In the bacteria, once you transform into the bacteria, you can do the selection process, because your vector must have some antibiotic resistance. So after on that particular against that antibiotic, you can select the colonies which have that vector, which have your gene of interest. So after that, you can multiply and select. So this is a recombinant DNA technology. One should be aware of this thing. So like in this topic, there are many things like vaccines. There are many types of vaccines. Like I've told you about 8 different types of vaccines, and there are different kind of hormones, pharmaceutical products. There are number of pharmaceutical products like growth hormones, insulin, antibody production. But in this talk, I'll just I will just give you the brief overview of like 2 of the vaccines and 2 of the pharmaceutical product. Like I will discuss about the hepatitis B virus vaccine and human papilloma virus vaccine using the recombinant DNA technology, and I will also discuss the production of insulin using recombinant DNA technology and the antibody production in the mouse. So as I told that recombinant DNA technology is can be helpful for production of vaccines and biomolecules. So this is the first example in the production of recombinant hepatitis B vaccine. So what we'll do? The thing is, we have to isolate a plasmid. We will make a cut in the plasmid with the restriction enzyme. This is a vector part, and we will take the hepatitis B virus strain, and we will take out the gene that is encoded for hepatitis B virus surface antigen. Okay, so hepatitis B virus antigen, we will encode that. We will ligate that sequence into the vector, into the plasmid where we have made a cut, and in the correct frame. After that, we will introduce that recombinant DNA into the yeast cell. Here we have taken the vector from bacterial cell, but we have to introduce this transform this into the yeast cell. Okay, so what will happen when we will transform in the yeast cell? Then we will multiply in the culture, and then we will able to select in the fermentation tank. There will be expression of hepatitis B antigen. That hepatitis B antigen we have to isolate with the downstream processing, and so we have to extract and purification, and then we have to get a quality control package. So that's how we can produce the hepatitis B vaccine. Now you have noted the point that you should note it that we have not used the complete virus. We are not using even the nucleic acid. We are just using a part of it, and it's work as a vaccine. So it is a quite safe. Production of Hpv vaccine. The thing is almost the same technology, same way. We have to take a plasmid. We have to encode the Hpv L1 site, L1 protein, so that we can have this VLPs like virus-like particles. So what happens when we will transform that in the yeast or insect cells? It will multiply over there, and then after multiplication, the encoding gene which have this L1 protein is also synthesized. So after and this will also get self assembling in the cell, and then after that we have to purify it, and that VLPs, if we inject in a human, it induct the antibodies and the CTL. So what happens when we inject it? It will protect from the HPV infection. HPV infection is most commonly in the uterine cervix. So it is, and it's a cancer causing infection. It can cause a cervical cancer. So it's better to be get it protected with the vaccine. The thing is, in most of the Western countries, this is a kind of mandatory vaccine. One has to get it. Now, I have discussed like two vaccine, hepatitis B and HPV. Now, this is a biopharmaceutical, like the production of insulin. What we have to do in the insulin for the production? The procedure will remain same. The standard procedure will remain same. Just we have to choose a different gene. The gene is a human insulin producing gene, and from where we can get it? We can get it from the human pancreatic cells. So we have to take the DNA from the human pancreatic cells, and we have to amplify the human insulin gene, and we have to tag it with the different restriction enzyme, and same way we have to prepare the recombinant DNA, like cutting and ligation and then transformation. So we have the recombinant bacteria. The recombinant bacteria, after selection, we can put in the fermentation tank, and then how the human insulin can produced. So recombinant bacteria multiplying and producing the human insulin in the fermentation tank. Then one need the downstream processing for extraction, purification, and of the human insulin, and further the packaging after the quality control check. This is the safest product. The diabetes is getting epidemic in many countries. There are so many people, millions of people are suffering with the diabetes, and diabetes also causes different kind of, it's kind of like start point of many diseases. Also diabetes affect the kidneys, also your eyes, your lungs, and your heart also. So to get a insulin which can really work well is very important, and it's good that this time many of the insulin products which are produced by the Recombinant technology are in the market, and they are the safest product. The production of antibodies, this is a little different than what we have discussed about the production of insulin and vaccines. To produce the antibodies, there is a little different procedure. Like we have to start from the point, then we have the antigen with us. So how to get the antigen you can get from the procedure I have discussed just now. Like recombinant DNA technology, you insert a gene that encode for particular antigen, and then you can get it expressed and isolate, and after that you have to inject that antigen into the mouse. Okay, because you have to produce the antibody, and you need a whole body for that. Okay, so mouse, if you inject the antigen, and after getting the antigen, the mouse and body will produce the antibodies. So you have to isolate the immune cells, and then get it fusion with the prepare the hybridoma, the tumor cells. And we have to screen for the production of antibodies. Antibody producing hybridoma needs to be cloned, and then further get it expand, and you can isolate the antibody. If one can also directly isolate, like if you inject in mouse, and you can take out the blood from the tail of the mouse, and isolate antibody, and from in the rabbit the antibodies can also be produced. So but main thing was we must have the antigen with us, and then we have to inject it to to induce the immune system, and once the specific titer of antibody is there, then you can isolate the antibody. Okay, thing is that with the emergence of this recombinant DNA technology or biotechnological tools, we got very nice kind of approach, and we can use it for development of many kind of vaccines, biological proteins, pharmaceutical proteins, hormones, and different kind of things, even without using the whole for production of vaccine. You may not need a complete microorganism. Okay, so That is the beauty of this technology. So let's summarize the things. The vaccination is a very effective approach to control the infectious agent, since even before the infection reaches you, your body has the antibody against it. So it will save you from any kind of microorganism, and if the proper vaccination is followed, then it means it's also good for the economy. Like it will remove the economical burden on the health sector, and that's why it's a very effective. And second thing, the vaccine is classified according to their formulations. How the vaccine are formulated, they are classified accordingly. Like as I discussed in different 8 types of vaccine, they are classified on the basis of their formulation, that how they are formulated, they are categorized in that way. And the third thing is that the cell and molecular tools will be the future for the production of vaccine and the pharmaceutical products. Just remember that, because these are very safer, and you can do in the laboratory. You can remove all the kind of chances of mishappening in the laboratory. You can remove all kind of genes that can be virulent, or they can even create any kind of problem. So that's why it will be used as a promising tool in future. And one more thing, that these techniques improve the safety and immunogenicity of the vaccines to a very high level, and production of pharmaceuticals also. So thank you.